Good morning, students. In today's revision part of fundamental unit of life, let us discuss the cell organelles or cytoplasmic organelles. What are cell organelles or cytoplasmic organelles? Membrane-bound specific functional regions in eukaryotic cells. Why the membrane-bound specific functional regions are there in eukaryotic cells? To keep the cellular chemical reactions separate from each other for their better efficiency. What are various cell organelles or cytoplasmic organelles present in most of the eukaryotic cells? Endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, vacuoles. These are present in all the cells, and plastids are present only in plant cells. So, let us discuss one by one. Endoplasmic reticulum. Endo means inside. Plasmic means of plasma or liquid inside the cell. And reticulum means network like. So the name itself is giving what it is. A large network of membrane bound tubes and sheets that looks like long tubules or round or oblong bags or vesicles is known as endoplasmic reticulum. The membrane of endoplasmic reticulum is similar in structure to the plasma membrane. The membrane of endoplasmic reticulum is similar in structure to the plasma membrane. There are two types of endoplasmic reticulums. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. Here ER refers endoplasmic reticulum. Rough endoplasmic reticulum has ribosomes attached to its surface, like how on a sandpaper sand granules are attached to the paper. In the same way, ribosomes are attached to the surface in rough endoplasmic reticulum. Ribosomes are the sites of protein synthesis. So, rough endoplasmic reticulum, it provides the surface for protein synthesis. The synthesized proteins are sent to different targets in the cell using endoplasmic reticulum provided vesicles as transport channels. Coming to smooth endoplasmic reticulum, it provides the surface for the biochemical reactions involved in lipid synthesis or fat synthesis. So totally, from rough endoplasmic reticulum, proteins are produced, and from smooth endoplasmic reticulum, lipids are produced. We already have learnt that plasma membrane is constructed more of proteins and lipids. Taking that point into consideration, let us learn one definition. Membrane biogenesis. Genesis means creation. Living membranes creation. The process of constructing or building the cell membrane using the endoplasmic reticulum synthesized proteins and lipids is known as membrane biogenesis. The process of constructing or building the cell membrane using the endoplasmic reticulum synthesized proteins and lipids. Are all proteins and lipids that are synthesized from rough and smooth endoplasmic reticulum respectively are going to be utilized in membrane biogenesis? No. Only some proteins and lipids are utilized in membrane biogenesis. And some proteins function as biocatalysts. What are these biocatalysts and all? When you come to class 10th onwards, you will understand. Anyhow, biocatalysts are also known as enzymes. All enzymes are proteins only. So some proteins and lipids, they are used in membrane biogenesis. Some proteins, they function as biocatalysts or enzymes, and some lipids function as hormones. So these are the fate of various biomolecules that are produced from endoplasmic reticulum. Functions of the endoplasmic reticulum. Endoplasmic reticulum, it serves as the channels for intracellular transport. Within the cell, various biomolecules are transported from one area of the cell to another area. So the channels that are utilized for this transport are provided by endoplasmic reticulum. Endoplasmic reticulum function as cytoplasmic framework which provides a surface for some biochemical activities. Already we have discussed protein synthesis, lipid synthesis. They are taking place on the cytoplasmic framework of this endoplasmic reticulum only. Example, in mammalian liver cells, smooth endoplasmic reticulum, it provides the surface for detoxification of poisons and drugs. So, due to various reasons, the animals may tend to take up certain chemicals into their body, knowingly or unknowingly. They will be having certain poisonous effect or toxic effect. If that toxicity is not removed, then they may cause harm to the body. So that removal of toxicity or detoxification of poisons and drugs is carried out on the surface provided by the smooth endoplasmic reticulum in liver cells. Next, Golgi apparatus. It was first described by a scientist known as Camillo Golgi, so the name was given Golgi apparatus. Golgi apparatus is a system of membrane-bound vesicles arranged parallel to each other in stacks or cisterns. Cellular membrane system. There are membranes in Golgi apparatus. There are membranes in endoplasmic reticulum. The membranes of Golgi apparatus and the membranes of endoplasmic reticulum have connections with each other and form cellular membrane system. Endoplasmic reticulum synthesizes various proteins and lipids. Such endoplasmic reticulum synthesized material is packaged and dispatched through Golgi apparatus. And what are the functions of Golgi apparatus? Just now we have discussed that endoplasmic reticulum synthesized material is packaged and dispatched through Golgi apparatus. Means whatever the material that is synthesized in endoplasmic reticulum, it comes out through Golgi apparatus. When this material is coming out through Golgi apparatus, certain processes are going to take place on these materials. What are they? Initially they are stored, and after some time they are modified, then they are packaged. So storage, modification and packaging of products in the form of vesicles is carried out by Golgi apparatus. And certain simple sugars, they are modified into complex sugars inside the Golgi apparatus. And the protein packets, some of the protein packets, they come out and are delivering their products at various targeted areas in the cell. Whereas some other protein packets or protein vesicles, they remain as separate cellular organelles known as lysosomes. So Golgi apparatus is involved in the formation of lysosomes. So lysosomes are the only organelles that are derived from the other cellular organelle within the cell. Lysosomes. Lysis means breakdown. Soma means body. Since lysosomes are the vesicles or the bodies that are involved in the breakdown of various biomolecules inside the cell, they are called lysosomes. Lysosomes are the waste disposal system of the cell. Whatever the waste that is created, it is taken into the lysosome, and inside of which it is broken down into smaller units. Then they are stored for a while in vacuoles. Later they may be discarded out. That's why lysosomes are known as waste disposal system of the cell. Lysosomes help the cell to be kept cleaning by digesting any foreign material and worn out organelles. Lysosomes, they help the cell to be kept clean by digesting any foreign material and worn out organelles. So many foreign materials enter inside the cell. Many of them may be not useful and some may be even harmful also. Such materials have to be broken down. They should not be kept in their complete structure. And worn out organelles. Many organelles, after they become old, they may be getting damaged or they may not be functional. Such organelles are known as worn out organelles. Right? Such organelles also have to be broken down into simpler units, because they will occupy unnecessarily the space inside the cell. So lysosomes help the cell to be kept clean by digesting any foreign material and worn out organelles. Foreign material like bacteria or food and old organelles, they end up in lysosomes and broken down into small pieces. Lysosomes can break down these bacteria, food, and old organelles, all of these above things which just now we have discussed. And because they contain the powerful digestive enzymes. So far we are discussing they are able to break down. How are they able to break down all these things? Because lysosomes contain powerful hydrolytic digestive enzymes. And actually, in fact, with hydrolytic digestive enzymes, whatever the vesicles that are coming out from the Golgi apparatus, they remain as organelles called lysosomes. And I just have told you, lysosomes are the only organelles that are derived from other organelles. Like how a cell is formed from the previously existing cell, lysosomes are the organelles that are formed from the previously existing other type of organelle. So lysosomes are also known as cells within the cell, right? During excessive cellular disturbance, lysosomes may burst out and the hydrolytic enzymes digest their own cell and cause the killing of their own cell. That's why they are called suicidal bags of the cell. Why lysosomes are called suicidal bags of the cell? Because during excessive cellular disturbance, lysosomes, they may burst out and the enzymes that are present in the lysosomes are released out and they digest their own cellular contents and cause the killing of the cell. So, lysosomes are called suicidal bags of the cell. So, finally, what can we say about lysosomes? Lysosomes are membrane-bound sacs filled with digestive enzymes. And these digestive enzymes are made by rough endoplasmic reticulum. So, what are lysosomes? They are the membrane-bound sacs filled with digestive enzymes, and these enzymes are made by rough endoplasmic reticulum. Next cell organelle, mitochondrion. It is singular word, mitochondrion. Mitochondria is plural form. Mitochondria are the powerhouses of the cell because energy that is needed for chemical activities for life, it is released from mitochondria in the form of ATP. ATP means adenosine triphosphate. And this ATP is known as energy currency of the cell. Whenever the energy needed for chemical activities, the terminal phosphate is broken down and the energy is released from the ATP. The energy stored in ATP is utilized by the body for making new chemical bonds and for mechanical work. Mitochondria has two membrane coverings, not one membrane covering. It has got two membrane coverings. The outer membrane is very porous, means many small pores are there, tiny pores are there in outer membrane, so as to allow certain materials to enter inside the mitochondria and certain materials to leave the mitochondria. And inner membrane is deeply folded. Deeply folded means, here you can see, this is one folding, this is one folding, this is one folding, this is one folding, this is like a deeply folded. These folds, they create a large surface area for ATP generating chemical reactions. We have just learnt from mitochondria ATP is generated. So these ATP generating chemical reactions are taking place in the inner membrane. If surface is less, lesser ATP generating chemical reactions can take place. If more surface area is there, then more chemical reactions can take place, right? And more ATPs can produced. So, folds create a large surface area for ATP generating chemical reactions. Plastids. As we already have said, they are present only in plant cells. Plastids are of two types, leucoplasts and chromoplasts. Leucoplasts are also called colorless plastids because they do not contain any colored substance or pigments. Chromoplasts, they contain colored substances or pigments. So, they are the colored plastids. The chromoplasts are the colored plastids containing chlorophyll are known as chloroplasts. They are involved in photosynthesis. These chloroplasts, they may contain yellow color pigment like xanthophyll or orange color pigments also. Leucoplasts, as I already said, do not contain any pigments, so they are colorless in appearance. Leucoplasts, they help in the food storage. Like starch is stored by special leucoplasts called amyloplasts. Oils are stored by special leucoplasts called elaioplasts. And proteins are stored by aleuroplasts. In internal structure of plastids, many membrane layers are embedded, means present in stroma of the plastid. Stroma means the inner area of the plastid that contains some liquid-like thing. In that liquid-like thing, coins like many membrane layers are present. Those coins like membrane layers are called grana. Both mitochondria and plastids, they are referred as strange organelles because they have their own DNA and ribosomes, which is the significant feature of only these two organelles. Other organelles do not have their own DNA and ribosomes. Because they have their own DNA and ribosomes, mitochondria and plastids are able to make some of their own proteins. Okay, in the next class we will discuss vacuoles and the cell division also. Okay, right? Thank you.